And that's going to be percentage. Now, I'll go back later on and put on there the actual value it stands for. It's still be what percentage is. All right, let me go back down. Let's work through this thing. Because you're going to see them again. My folks know I'm going to make a test out of this paper test. Roger and Wendy Wanda doesn't know that. One day they're going to come back and hit you as a paper test. I do like 4 at a time, my 4 lessons. This is my first lesson test. I'm going to put 4 together, run them off, and do a test on them. So you see them one time, you'll see them again. And I usually add a few things to it to make it worth your while, make it fun.
Can I take
most of them you've seen. I don't add any of them.
The pictures?
I want to change this right now, so that when I ever do it again, people will have to, they can number and make their plans there. I'm going to save that thing.
It's right there.
That's how they'll number in the future. All right. Well, what did you say on this one?
Photosynthesis.
Process of
Photosynthesis.
And then those that do it are called
Autotrophs.
autotrophs. We're heterotrophs. So we eat different. Okay. Now this one, all that stuff I said was to give you information, but the bottom line is, what is the name of the green pigment that's not
chlorophyll?
Chlorophyll. It's chlorophyll. Now I could have asked a question, and I might modify it. A green pigment blank found in organelles called blank. What did you say then? The green pigment chlorophyll found in organelles called. What do you find? Chloroplast. Chloroplast. So I do things like that just to add a little more interest to it. For these two, what is a plant splitting to get those H's? He's splitting water. So what's he going to throw away he don't want? That we need? The oxygen. He splits the water. He wants the hydrogen. He uses those NAD, FAD scattered. And the oxygen, he throws away. Thank God he does, because I didn't use that. So it's water and ions of oxygen. And my clue right here, I thought would give you that. You got to have live, that's oxygen. So when you have H's and O's, I would think of water.
So 5 is oxygen, right?
Huh? And 5 is oxygen.
Okay.
But you breathe oxygen.
So if you put hydrogen and O2 there, is that
What did you put before?
hydrogen?
No, you ain't splitting hydrogen. When it is split, that refers to number 4. When water is split, then hydrogen comes off. Hydrogen is used by the plant. The oxygen he has no need for, and he gets rid of it as a waste product that gives me my life. So 4 can't be hydrogen if you split the 2. You can't. You can't split hydrogen. You get hydrogen. See that word "it" right there? This is pointing back to number 4. When it is split, you can't split hydrogen and expect to get hydrogen too.
I think I combined
What is split?
See, when it is split refers back to what number 4 is.
Energy.
Because if I'm doing a lecture, I would say uses water when water is split.
Right.
Now, I can't do it here. I made blanks on it. I had to use the it. What do you call the thing that actually grabs hold to and hangs on?
Acceptor C.
It's called acceptor. C. All right. One of the acceptors is called NAD, and it's a coenzyme in all living cells. And what's it carrying, Hillary? What? It is carrying hydrogen. And then, by the way, what's hydrogen carrying?
Electrons.
Electrons. But he is grabbing the hydrogen. He can't grab the electrons, too little.
Would you look and see what I picked for that? 'Cause I can't even remember.
When you get it back, you'll know. Make a note to check out your number 7.
Number
Now,
7.
The same question came up with this one, but I'm using FAD. It's the same answer again. He's grabbing the hydrogens. The electron. You know how big an electron is?
It's very tiny.
If you take a proton and you cut it into 1,000 835 pieces. One of them is the size of an electron. He can't grab an electron. He can grab a proton though, but the electron is too little. Like you couldn't grab one atom of oxygen, but you can grab the air.
You gave a hint. You said the other electron hydrogen acceptor. You're telling us that it's the hydrogen acceptor in the question.
Well, I'm saying it's the acceptor of both of them. But while I did word it that way on purpose, so these two would fall together. I did the same way. I could have said hydrogen slash this acceptor. That'd been even more confusing. That'd been mean. So when I had it worded, I want you to associate these together. That was the idea, and that doesn't work. But I could have reversed those two and still call for H because it's just too little. You can't grab it. But you can grab what's carrying the electron, and that's what they do. And when you split the water, those hydrogens, you gotta grab that sucker. Plants don't let go of hydrogen. They let go of the other half of water, which was the oxygen. All right. Three of those are flat out right, and one of them is stupid.
It's wrong.
It's not stupid. It's wrong. It's not stupid.
It's wrong.
It doesn't use oxygen. It does not use oxygen. That's the beauty of glycolysis. You don't use oxygen until you go into the mitochondria, and this happens in the cytoplasm where you don't need oxygen. So A is not right. Let me look at that one too. All right. Number 10. Oh, all three of these. Okay, now. Glucose is hydrolyzed and it makes enough energy to make how many brand new ATPs?
4.
4. It took two to make it happen. So when you get down and look at the differences, you've only made a net gain of 2. 4 to 2. Now I can rewrite that bad boy, and I probably will when I do the real test. Because I don't want you to memorize it, I want you to know it. What I got to do is reverse those and a little bit of wording on my part, and I'd have number 12 be number 1, and go backwards to it. So your best, pictures are nice, but your best actually never stop. All right. Very top, what do you have? Is glucose. And those two red boxes, same word in both of them, but what's that word? I'll either take pyruvate, or I will also take pyruvic acid.
Okay.
They're the same words.
Okay.
So actually, this is 6 carbons. There's 3, and there's 3. Still got 6, but now they're in 2 things.
So,
Yes,
I left
ma'am.
off the word acid.
What now?
I left off the word acid.
What did you say?
Pyruvate.
If you say pyruvate, you better apply the word acid. If you said pyruvate, I won't mark you all wrong, but you're not all right either. So if you want to leave the acid off, you got to say pyruvate.
I said pyruvate.
If you can't spell pyruvate, you should spell pyruvic. You better add the acid word.
I said pyruvate.
So I'll find out what you did. But the answer is either pyruvate or it is pyruvic acid. Now, when it's over, the presence of what? Oxygen determines what happens next. Now, if you have it, then what kind of respiration occurs?
Aerobic.
It's called aerobic respiration. If you don't have it, then what happens?
Fermentation.
Fermentation.
You saying anaerobic?
Now I'm saying aerobic. And means no.
Without oxygen.
Yeah. If you say AER, that's with it. ANAER means without it. Okay? Well, by itself, aerobic respiration produces how many?
36 molecules.
36. Now, if you tack on glycolysis, which is 2, what's the grand total?
38.
38. 36, 38. Okay? Now, when aerobic is complete, the glucose is gone. It's been all broken to pieces. And what do you actually have left over? The order's not important. What do you have?
Water.
You have carbon dioxide, and you also have
Water.
Yeah, water. It's right there.
Water.
And when out comes carbon dioxide too. So I don't care. I put down probably water and carbon dioxide, but you can reverse those two.
I wrote water, I crossed it out, I put H2O.
Well, that's water.
Okay.
I'll accept that. I'll accept H2O.
I know that seems simple, but
I ain't gonna write the two words carbon dioxide. So I now said
You have
CO2.
to
I
choose
mixed them up.
to, yeah.
I got a lot of stuff marked out and written like that. Just
As long as you change it to the right answer or just another right thing. Folks who change it wrong, they get mad at themselves. Should have left it alone.
Happens
So we all do that. We can't help it. We can't just leave it alone. When glycolysis is over, but there's no oxygen, fermentation happens. And what are the two kinds of fermentation?
Lactic acid
Lactic
fermentation,
acid, and then
ethanol or alcohol.
Okay, I'll take alcoholic, I'll take ethanol. You can reverse those two. So I'm looking for just that they're there. Well, what kind of fermentation it depends on the metabolism, and that depends on
Enzyme.
the kind of enzyme he's going to use.
Oh, I guess that
The enzyme makes a difference. Yeah. What'd you say?
So I guess that one right.
Oh, you what? What is it going to be?
Well, I couldn't think of it, then you moved on,
And it
and then
said
I couldn't
enzyme is the
remember
next
it, so I
question.
just said enzyme.
Yeah,
Good idea.
to the next question. That's where I got my answer.
Oh, the next one gave you the answer? Okay, let's see what it.
When you answer.
All right. Obligate anaerobes. There's an anaerobe. That means you can't give them oxygen, you'll kill them. Okay. Like some
Bacteria and yeast.
and
Bacteria and yeast.
you can mix them up.
Yeah.
I know those two will die if exposed to oxygen. For these organisms, they got to do fermentation. And they do that to make what? To make ATP. They must have the ATP. It all boils down to ATP. And you need to use that to make new organisms, whatever. So
from this point on, my memory card is full.
the way I did it basically was I just
Full.
said yeast and bacteria, knowing full well you could verse those two. And then I came back and said
Okay.
ATP. All right. Lactic acid fermentation occurs mainly in
Animals.
animal
Animal
muscle.
muscle.
When you run real hard and you can't make enough oxygen, you're going to have that going on. Now there's that enzyme that might trigger your thought. And now, which of those did I not make up?
C.
C is for real. C is for real. Okay. The other two, I wanted an LD and there's no H in that word. I just realized that's a no. That's stupid. That's these two. I don't know where I got that word from. I just threw letters on screen. And this one, duo, sounded like two. That sounded good to me.
Yeah, 2 hydrogen.
That's the answer right there.
Yeah.
Okay. Now, the lactate formed by lactic acid fermentation can be recycled by what organ?
Liver.
Which two can you kick out right now? The pancreas too. Get rid of it. That's why I said back into it. Now you're concerned with A and B, right? Now, you keep back. Okay. In the liver, it is changed. Is it changed to glucose or is it changed to pyruvate?
To pyruvate.
It's got to go to pyruvate so it can then go back to
Glucose.
right. So back in, you're going to realize that, well, it cannot be A because you can't do glucose to pyruvate. It's got to be B.
B.
So I would have backed into B
Yeah.
if I didn't know it for sure. And I would first, I get rid of C and D and give me a 50-50 chance. And I keep backing and keep trying to realize what cannot happen. And you ain't going to have glucose then going back to pyruvate.
And then carry
That's
that
right, back where you
blood
were. It doesn't happen.
to the organism.
There's only got to be
The conversion of blood. Conversion. D.
Oh.
Okay. Well, now, this changing, well, right there, you see right there the answer to the previous one?
Gluconeogenesis.
Now, when I do a paper test, you get to look back, don't you? So that's the advantage you have, is when you go, oh, I think I know an answer one, then you got to find that one in all the papers. But anyway, all those are good, but which one really is the conversion?
D.
Gluconeogenesis. That's a new start. New start. Okay. Well, there's your fermentation is ethanol or alcohol. I use both interchangeably. Now, all what do this?
Yeast.
All yeast do it, and some
Bacteria.
do it. Now, when they get done, they have the ethanol, the alcohol, but what else do they have they give off?
Carbon dioxide.
Carbon dioxide. Because a baker needs that for the bread. And the brewer needs it for the liquor. Okay? All right? In the two industries, obviously.
Baking.
I said bakeries. If you said baking, you're fine. I said bakeries
And
breweries.
brewers.
I said breweries or brewers or Schlitz or Budweiser. I understand that them folks have that stuff in a lock and key too. Those yeast they're using, they guard that with their life apparently. It's really secretive, like Coca-Cola formula. All right. Now, if you have oxygen, then you're going to do aerobic respiration because oxygen is going to be the final acceptor of the electrons. When they get done, they got to be cleaned and taken care of. Well, they move down to ETC until they finally get done, and the oxygen grabs them and goes out with them. But what does ETC stand for?
Electron transport chain.
Transport chain. And that came up on the test later on too, so you can verify you're right. Most folks get it right here. And the other slide just verifies it. All right, what was that?
Mitochondria.
How'd you say it?
RION.
Mitochondrion. But mitochondria would be another one on there too. Mitochondrion is one. And those things right there, I know they're cristae, but is it TA or TAE?
TAE.
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 The E means a lot more than one. And when I do it, it's just a bigger word. Krista is a small word. Krista, it's a Krista, must be and it works that way.
I don't know if I wrote the E. Did I cross it
So you
off?
went back and forth. All right. There's a little bit of ATP produced during the Krebs cycle, but I want to know what's the Krebs cycle also called?
Citric acid
It's
cycle.
a citric acid cycle. Now, if I'd asked why, would you know that? If I'd asked why, would you know that answer?
What?
The reason why is it called a citric acid cycle?
Is the first product produced in the cycle is
Exactly.
citric.
The first product produced in the cycle is citric
Citric.
acid. I mean, it was called that before it was called Krebs.
Go girl.
But she's cooking.
Yes, write that out.
Her hair's not pulled too tight like hers. Now here we go. All right, now we're going to break it all down. Glycolysis by itself makes how many?
Two.
Okay. The Krebs cycle makes how many?
Two.
Two also. There's your two twos. All right, now. The ETC makes
34.
34. So just the respiration part makes
36.
When you add them all together, you have
38.
You got it right.
I'm gonna tell you what stumped me. When it came back, I don't know why I thought about 44 and 45. I'm like, those are not supposed to be the same. Those are two different things. But then I had to read.
Wait, what was it
Because you
again?
used two twice.
What was it again?
I don't know what I said.
2, 34, 36.
38.
Yeah, I missed those.
And you missed all of them?
No, I didn't miss all of them, but I.
Oh, good.
I got one right. The first one. Because I heard them.
Well, that's positive thinking. Now, you'll see them again, and I might have these things broken up all over the test. 41 might be here, and 42 might be over here, and I might not have them so you can see them. I think seeing them helps you do them, because you can see how they line up. But on a paper test, I won't have them like that. I'll have number 1 might be the first one, and then 25 might be the second one, and 30 might be the third one, so be aware. It won't be as easy if you call this easy.
A.
Which one did I make up?
B.
I made up B.
Hey, A is the answer.
A is the answer.
Yeah, B makes no sense because you can't produce ATP before you do glycolysis.
That's exactly right. That's exactly right. But all the words, it just look like it.
Standby.
Yeah, fermentation
Mind for standby.
actually does nothing. Good thing it doesn't. The main thing about fermentation, so you should have asked, and you didn't, you should have asked, well, if fermentation gives you no ATP, why is it doing it for? What's it doing it for? If fermentation gives you no ATPs, why not just stop there and forget the rest of it? Well, your problem is, you got 2 pyruvic acids. You gotta get rid of them. You gotta get rid of pyruvic acids to do it again. And when you get rid of pyruvic acids, when you get done, you have ethanol and you have carbon dioxide. All that yeast did was make room for more glucose. So I can then make more pyruvic acid. I can't stop there because now the pyruvic acid is in the way. I got to get rid of it. I do fermentation to get rid of it. So when fermentation is over, he's back to more glucose. And that's because I asked that question one time when I was a student, and I said, as this professor, if the fermentation is not making any ATPs, why even do it? And he said, Mr. Ted, you got to get rid of the pyruvic acids. I remember that conversation this very day. We talked in class like I was somebody. Some professors talk like you. And he says, you know, and I hate it when they say, good question. You know what I'm talking about? Because I know what I asked it. He didn't say that. He says the fermentation is an avenue to get rid of the pyruvic acid so you can then do it again. As long as the pyruvic acid is there, then you can't start and make more ATPs and the yeast will die. And when I took the little slide I had, I was going to look at under my scope at home, I thought was dust. Those are yeast. And I went, my God, these things are little bitty. But they make a lot of carbon dioxide in the bakery, and they make a lot of alcohol in the brewery. But that's the reason why. Because when he first explained that, my mind was going, well, that's wasteful. Why do fermentation? It's not good for anything. He said, but it is good for something. You got to get rid of the pyruvic acid so you can make room for more glucose and do it again. So, oh, okay, that makes sense to me.
Last one.
Which one's the chain?
B, bravo.
Yeah, this is chain. That's the chain going across.
That's right. I'm good.
That's the chain.
Oh, that's very
Well, I'll
well.
check these tonight, and I have a lab coming here at 5:30, so